to lecture 2 of enzymes, and today the topic that we have is lock and key hypothesis. The lock and key hypothesis is basically a very important topic when it comes to enzymes in Olevels, and in this topic basically we need to remember that what is a lock and what does the key represents. So the one thing that we need to remember is this lock and what does the key represents. So the lock represents our enzyme and the key represents our substrate. As we can see, when the key combines with our log, so basically in turn it forms a product. That means when an enzyme is reacted with a substrate, it forms an enzyme-substrate complex that in turn convert into a product. Since the product can be multiple or it can be singular as well. So this is a technique that can be very helpful for us in solving some MCQs if you remember that what is a lock and what is a key represent. So for example, have a look at this example that says when a protein is converted into amino acid, let's say in presence of protease, and the question is how can we determine that what is a lock and what is a key? So the very simple way or the phenomena from which we can determine is that the lock is always an enzyme. An enzyme can be represented or enzyme can be determined by the fact that they have A's in their name. So since protease react on protein in order to convert it into amino acid, protease in an enzyme, that means it is a lock. Whereas protein is a substrate that in turn forms a product that is an amino acid. So it is very simple to be understood that when a protease acts upon protein, it forms a product that is amino acid, and this helps us also understand that what is a lock and what is a key, and how it determines that what is an enzyme and what is a substrate. This is all we need to learn about lock and key hypothesis. The next topic that we have for today is how we are going to learn about some simple facts about enzyme. We're going to discuss enzymes overall, like not in such very detail, but facts that we always need to remember about enzymes. Number one fact is that enzymes are biological catalysts. Now, what is meant by biological catalysts? Enzymes are biological catalysts that basically speeds up a reaction without being involved in it. Enzymes is not involved in a chemical reaction. However, they always remain unchanged until the end of a reaction. Okay? And the next point that we need to remember that they are proteinous in nature. We have often heard that enzymes are proteinous in nature. Now, what is meant by proteinous is that they are made up of protein. That's why we say that enzymes are very proteinous in nature. The third thing that we need to remember is that enzymes are very specific. When it comes to a substrate, does not allow all the substrate to be attached to an enzyme. So only, for example, protease can act upon protein, not lipase. Similarly, lipase cannot act upon protein. It can only act upon lipid in order to form a product. Alright? So the next thing that we need to really learn about it is what is meant by the term denatured when it comes to enzyme. So the denatured term is basically referred to when it comes to a temperature. Whenever the temperature is raised above the optimum temperature, what is meant by an optimum temperature? Optimum temperature means any temperature at which the enzyme works best. Every enzyme has a different optimum temperature. So whenever the temperature is raised or being very below than the optimum temperature, then the enzyme gets denatured, that it does not perform its action very efficiently. It becomes very inefficient. So these are some of the very small points that we all need to remember about enzyme. And lastly, we are going to discuss how we can name an enzyme. So the last topic that we have for today is naming enzyme. This is a very simple process, as we all know that enzymes can be named we need to remember that we can be named on basically If anything that have a A's in it, it means it is an enzyme. For example, let's say lipase, let's say protease. We have taken these example multiple times, so let's take another example. For example, peptidase. For example, maltase or sucrase. So basically these determine that whom they are going to act upon. For example, sucrase will basically act upon sucrose, maltase will be acting upon maltose, peptidase will be acting upon peptidose, and similarly protease on protein and lipase on lipids. So simple it is that naming an enzyme means that it is going to consist of the word or the term is in it. If you're going to determine and given us the options about four option that which is an enzyme, we can similarly easily determine it by is. Whatever has A's in it, it is an enzyme. This is basically a very simple phenomenon of learning how to name an enzyme, as we have discussed. What are the simple facts about enzyme? That they're biological catalyst. They can speed up a chemical reaction without being involved in it. They're very proteinous in nature and they're made up of protein. They're very specific. They does not allow any enzyme to be attached to it, and they can also become very denatured if the temperature that they're provided is not optimum. And the first thing that we actually discuss is lock and key hypothesis, that we need to learn what is a log and what is a key. This is a trick point that you all can remember, is that how you're going to learn that log represents that it is an enzyme and key represents it is a substrate. So it will be easy for all of us to solve any equations. That's it.